For exercise 1, we are going to use Benedict's reagent to detect reducing sugars in a variety of substances. So we've got to prepare our substances. And the first thing you're going to do before you even prepare anything is take a look at data table 1 and these 9 substances listed and predict whether these substances are going to be reducing sugars or not. Now, it says here it doesn't matter if you're right or not, and that is true, but if you've read the chapter and if you've done the homework, you should have a sense of whether some of these are going to be reducing sugars or not. So use your common sense and use what you've learned in this section to predict. All right, and then the next thing you're going to do is you're going to create test solutions for table sugar, Splenda, and Sweet and Low. You're going to gather 3 test tubes, and you're going to put a ruler next to those test tubes, and you're going to make a mark half a centimeter from the bottom and 7 centimeters from the bottom. And you're going to do that for 3 test tubes, and then after you've made those marks, you're going to label each test tube according to the solution that you're going to make. So SUG will stand for sugar, SPL will stand for Splenda, and SNL will stand for Sweet and Low. Then you will fill each test tube to the half centimeter mark with each solid substance. So take your table sugar, add it to the half centimeter mark, the Splenda, add it to the half centimeter mark, and then the Sweet and Low and add it to the half centimeter mark. Then you're going to fill up to the 7 centimeter mark with distilled water. So you can do that using a pipette that you'll label DW, fill it with distilled water, and add the distilled water to each of these test tubes. And you'll use this distilled water pipette, this short stem pipette, throughout the experiment, so don't use it for anything other than distilled water. Next, it tells you to place a clean gloved finger over the top of each test tube and invert it or turn it upside down to mix the solid until it is completely or at least mostly dissolved. It's going to be really, really important that either you don't use the same finger that when you put it over and turn it upside down of the glove, or that you wash it very carefully in between each inversion, because when you turn this upside down, some of the solution is going to get on your glove, and then if you use the same finger, that solution will get into the other test tube, and then your results will not necessarily be right because each solution or 2 of the solutions will be contaminated. So be sure that you use a different finger, gloved finger, for inverting it, or wash your gloved hand carefully between the 2. So those will be your 3 solutions that we'll use a little bit. And then you want to prepare your potato. You want to cut a little piece of potato about half a centimeter cube, so half a centimeter on each dimension, length, width, and height, and you can use your ruler to determine how big that is. And then with a sharp kitchen knife, cut that potato into really, really, really tiny pieces, and then use the back of the spoon to crush it even more until you have sort of a pulp. And then we will use that in just a little bit to create a potato solution. All right, so then you want to gather 9 empty test tubes and a marking pen. Lay the ruler next to each and place a mark a half a centimeter from the bottom of the test tube and 2 centimeters from the bottom of the test tube. And then label them 1 through 9. And this 1 through 9 designation is going to refer to what's labeled in what is listed in data table 1. So number 1 is just distilled water. Test tube 2 will hold glucose, 3 will hold starch, 4 will hold table sugar, 5 Splenda, 6 Sweet and Low, 7 milk, 8 juice, and 9 potato. So after you've labeled them and put the 2 marks on them, then you're going to fill each test tube up to the half centimeter line with its corresponding substance. So for test tube number 1, get your pipette that you labeled DW and add distilled water up to the half centimeter mark. Okay, and then for 2, glucose and starch solution, those are in, HOL has provided those, so those should be in your macromolecules experiment bag. 4, 5, and 6, table sugar, Splenda, and Sweet and Low, you have made those solutions, and you should have pipettes in these test tubes, so use those pipettes for each individual solution to put it in 4, 5, and 6. And let's see, and then for milk and juice, carefully add those so they go up to the ½ centimeter mark. And then for the potato, what you're going to do is you're going to gather a little bit of the potato, stick it into the test tube number 9 until it gets up to the ½ centimeter mark, and then add water if you need to. There might be enough juice from the potato to get it up to the ½ centimeter mark, but if not, add some water. Alright, then, now you've prepared your 9 solutions. Now you get your dropper bottle of Benedict's reagent and add Benedict's reagent to each test tube up to the 2 centimeter line, and then swirl the test tubes, put them back in the 24 well plate to hold them, and then you're going to make a hot water bath. So partially fill a 250 milliliter beaker with tap water that is just under boiling water. So while you are preparing your solutions, you can put a pot of water on the stove, or you can also put water in the beaker and then put it in the microwave until small bubbles begin to form. And if the water's on the stove, you can just boil it and then turn it off, and when it's stopped boiling, add it to the beaker. And then you're going to let the test tubes sit in the hot water bath for about 5 minutes. If they don't all fit, just put 5 in it at first and let it sit for 5 minutes, and then remove them and place the remaining 4 in the warm water bath or hot water bath for 5 minutes. And be sure to handle the test tubes with clamps because they are going to heat up and you don't want to burn yourself. Alright, so once the incubation is complete, once they've warmed up for 5 minutes, when you are removing it from the hot water bath and putting it into the 24 well plate to hold it, swirl the contents thoroughly to mix, and then record the final color in data table 1. So here's where you place the final color, and then the test result for the reducing sugar. There's a color-coded key in your exploration section of this lab to tell you what color means what with regard to reducing sugar, how much reducing sugar. Alright, and then you want to clean the test tubes with soap, tap water, and a test tube brush. Everything that's in those test tubes can go down the sink. You don't have to take off the markings or the labels. Do not dispose of the sugar, Splenda, and SNL test tubes, and the unused potato, the unused juice, the unused milk. Don't dispose of those. Just dispose of what was in the test tubes labeled 1 through 9. Okay, so looking at exercise questions 1, you should answer question 1, table sugar is sucrose, and then explain the science behind your results. Well, you can use your textbook to answer this. It explains why sucrose is or isn't a reducing sugar. So the science behind it just means why is it not a reducing sugar. Do Splenda and Sweet and Low test positive or negative? So answer that. List the ingredients found in each of these products. So look at the packet to do that and underline the ingredient that is the artificial sweetener. So this is reviewed in the exploration section of the HOL lab. What is the relationship of glucose and dextrose? Don't answer question 3, and don't answer question 4, and don't answer question 5. So for exercise 1, all you have to do is answer question 1 and 2.